And one of the questions we wanted to know the answer to is, what is the cell of origin of prostate cancer? And there's a lot of controversy in the field. There's a dominant school of thought that says that it's a luminal cell. And the majority of that thought comes from the fact that human prostate cancers express luminal markers like the PSA antigen. There's also a very nice paper that came out from Koryabate Shen and her husband Michael Shen, shown here, in which a specific gene, NKX3.1, can be marked by a knock-in experiment. And those cells which are marked in that fashion, some of them can survive the insult of androgen withdrawal, and then later show that they can turn into a cancer if you cross them with a P10 mouse or P10 loss of function mouse, where that occurs in a certain stage of prostate development. So there's at least a subpopulation of luminal cells defined here that can be a source of prostate cancer in the mouse. Other thoughts are that there's certain cancers have sort of ambiguous markers, both basal and luminal markers, and then finally there's some evidence that basal cells might be the origin. First of all, some tumors contain rare basal cells. There's both luminal and a neuroendocrine cell type that can occur in the cancers, et cetera, and it's shown here. And so we decided to test this more directly. And the style of experiment, again, is to take populations of cells, separate them cleanly into basal stem cells or into luminal cells, introduce various oncogenic insults, and evaluate the outcome. And the take-home message, and I'll show you 3 examples here, but we've done it with a dozen different genes and multiple combinations, is the only cell type in these assays that we can transform with high efficiency are the basal stem-like cells that are the same cells that can regenerate the tissue. And it works with ERG alone, AKT alone, or if we use paracrine stimulation by putting an FGF secretion into the stroma cells. Now, that doesn't say that it's the only way to make cancer, but it certainly says in this system it's a dominant way to make cancer. And I suspect both cell populations under certain conditions can serve as targets. If that's true, if the basal stem cells can be an important source for cancer generation, what controls their growth? What controls their self-renewal? Well, one of the things we decided to look at was that collection of pathways in which embryonically regulate cell self-renewal and are maintained in adult stem cell populations. There's a lot to say about this, but we focus particularly on the polychrome group for a number of reasons, and I'll tell you some results of work of Rita Lukács, a graduate student in the lab, not yet published, but I think of interest. And this take-home message is that the polychrome group of proteins, in particular BMI1, has a clear correlate with prostate cancer development and progression. So she did this experiment to test if it was essential for self-renewal of prostate using a knockdown strategy with a very good shRNA, again, isolated cells, placed into this prostosphere assay. And the take-home message is that if you inhibit BMI1, you get a very dramatic reduction in the number and the size of the spheres. If you use a form of BMI that can't be knocked down by the shRNA, you can reverse it. But maybe more interesting is to ask whether or not this pathway, which is known to occur in many tissues, had any particular dominance or predominance in the self-renewal. And so we used the second mechanism to enhance self-renewal, which is a Crelox-mediated deletion, which would stabilize beta-catenin, one of the other important players in the Notch pathway. And what happens is quite interesting. If you delete and make that deleted form of beta-catenin, you enhance the ability to form these spheres. But if you doubly infect them and now block BMI1, even in the face of augmented levels of beta-catenin, activated beta-catenin, you can suppress the self-renewal activity. So there may be, I'm not saying that it's completely dominant over all other self-renewal pathways, but there may be dominant or predominant self-renewal pathways in the prostate that are a focus for new targets for therapy. And then finally, well, if it's important for self-renewal, does it have anything to do with hyperplasia or cancer phenotype? And so this type of experiment can be done. Here, using either the BMI1 shRNA or control scramble, we can introduce that into cells and then plate them in this in vivo regeneration in the face of a large excess of fibroblast growth factor 10, which we know will induce a very hyperplastic and progressive towards cancer phenotype. And the important point I think you can see here is if you don't block BMI, then the cells respond with this hyperplastic phenotype. If you do block with BMI, the cells go back to a much more normal phenotype, and using the link color marker, those tubules which look normal are the ones that are red because we've knocked down BMI1. So again, I think a reasonable place to look to develop new therapeutics for prostate cancer. All right, so all of that study that I showed you was in the mouse, and I'm a firm believer in mouse models. I wouldn't dare come here and say anything else. But I also think that there are times when you need to look at human tissue and the specific features of human biology to ask questions about oncogenesis and particularly therapeutics. But we ran into a real snag. The markers we had used to isolate these tissue stem cells from the mouse included SCA1, and SCA1 unfortunately isn't expressed in humans. So Andrew Goldstein, a student in the lab, went out to find new markers with which we could fractionate the human prostate tissue stem cells. He found several, one of which called TROP2 from this microarray type analysis emerged. And I'll just show you one piece of data from his paper on this, which is to say that cells isolated with the phenotype of Lin-negative, SCA1, CD4 positive, TROP2 high are really the best stem cell population that we found. They can be put in, those cells isolated can be put into this in vivo system. They give the highest number of tubules with all the right structure, et cetera. Fortunately, that TROP2 marker is expressed in human, and interestingly enough, it's already been used is considered as a target in prostate and some other epithelial cancer therapies based simply on its tissue distribution and the fact that several companies have made very good monoclonal antibodies against it. Its function is really unknown. So we set out to see if we could not only repeat what we had done in the mouse and human, but add some new wrinkles to what we could study. And the outline of what we do is here, and I just have to stress that this type of experiment is probably more difficult at the doing level than the conceptual level by multiple orders of magnitude. We have very good collaboration with our surgeons, our pathologists, including Jyoti Wang and other labs like Isla Garraway's with which we share the tissue, because I think each tissue sample we get is precious and should be used to maximize research for the benefit of all. So we've gotten this pattern down. We were able to get benign tissue. Very importantly, we can clearly separate out the benign from the malignant tissue on frozen sections and alternate pieces of the biopsy, and do the following type of experiment, which is to see if we could purify the human basal cells, epithelial stem cells, away from the luminal cells. There's a lot of data here, but I just want to say that the criteria to define basal versus luminal is largely based on a co-linked set of markers, almost all of which are intracellular. So in order to show that, you have to separate populations and do Western blots, PCR, and in this case, intracellular FACS for the right cytokeratins. And all that was done, and I can tell you that we have a very clean basal stem cell population separated from the luminal cells. When we take those cells and ask which of them are capable of giving rise to regeneration, the answer, which of them are able to be infected with lentiviruses and then survive that, the answer is both the basal and the luminal cells can be infected, and both of them can survive for reasonable periods of time. So we're not looking at a selective loss of cell viability. When we use those in the regeneration assay, it's very clear that the basal-derived cells are the only ones that can give rise to these tubular structures. Here's that, for example, here, and in comparison, an actual histological sample taken from the benign region of a patient. Again, the tubule structure, all of the markers, fits very nicely, so we feel quite confident that we're taking a population of cells able to regenerate a tissue-like structure. And so finally, the experiment, can we take human cells and directly transform them with the same oncogenic principles with which we had done with the mouse? Now, this turns out to be an interesting issue. If you step back from it and realize that there are very, very few examples in which human epithelial cells have been directly transformed. Most of the time, one needs to worry about telomere length and add, for example, the catalytic subunit of telomerase. One needs to worry about some sort of self-renewal pathways and cell cycle blockade. Frequently, you'll see cell lines derived adding large T antigen or other manipulations along those lines. But we thought that this tissue regeneration system and the stem cells might not need to use those tools, and we could go directly in a single step from naive cells out of a patient with these genes to an oncogenic phenotype. And so the strategy here was, because we anticipated it wouldn't be so simple, we put together several genes, including AKT and ERG, which I previously showed you was an effective synergen in transformation, take the cells in this basal region, or the others if we needed to, and then infect it with this lentivirus and plate into the immune defective mouse. The answer is really quite pleasing. Here you can see basal-derived tubules coming only from the basal cells, none from the luminal cells. And these have achieved the histological structure that we'd refer to as PIN, really indistinguishable from the PIN lesions one would find in a human patient. And here's all the markers that goes with it. The important markers is that they're AR positive, P63 positive, to a certain extent, PSA positive now, that critical human marker that delineates the prostate lineage, and a gene called AMACAR, which is associated with the early progression, and so on. Well, if we could make PIN, could we make cancer? And to do that, we made a 3-gene combination. In this case, the same virus that had the AKT and ERG, but now adding another gene that has androgen receptor, because in every case we've tried, higher levels of androgen receptor are correlated with an enhanced progression of the phenotype. And this result is extremely pleasing to us because we can take those basal-derived human naive epithelial cells, add this combination of genes, and drive out lesions which are indistinguishable to our sets of pathologists from those found in natural clinical human prostate cancer, including expression of PSA, this AMACAR, AR, and a low level of P63. If you look even further, and this is sort of the 30,000-foot level, one of the hallmarks of prostate cancer is this variability, this different progression of phenotype and different multifocality. Here's an example from the regenerated prostate cancer that we're making in the kidney capsule of these mice. Here's a clinical prostate cancer specimen. All the arrows you can look through on your own, but the important point is that the phenotype that one should see between normal, PIN, and cancer with regards to these markers, that we can find the entire range of these phenotypes in our regenerated system. And in a sense, the system is looking in some ways like the variability you get back from normal human prostate cancer when you take biopsies from multiple regions of the prostate. So I don't think there's any doubt in my mind that at least basal epithelial cells can serve as a site for genetic damage to give rise to both PIN and full prostate cancer. And these cells defined by these surface markers should be very useful for both gene discovery, be it messenger RNAs or microRNAs. It should also be very useful for testing new concepts of oncogenesis. And I think most importantly, since they're human, for those human-specific therapies like monoclonal antibodies or enzymes for which drugs are significantly different between the mouse and human species, I think provide a very useful tool, not for the discovery of new therapeutics, but perhaps for their testing preclinical trial. I won't go through all the names of the people who did the work other than to thank them because I try to mention them during the talk. And thank you for your attention.